I am going to introduce you with the term chromosome. What is chromosome, where it is situated, what its function, etc. etc. You have already learned that chromosome is present inside the nucleus of a cell, maybe plant cell or animal cell. What is its function? Its function is that all organisms can pass its genetic character or trait generation after generation among their offsprings. So, chromosome is the unit, chromosome is the physical carrier of heredity. So, let us start with a diagram. It is a cell and this one is a nucleus. Inside nucleus there is nucleoplasm, and within the nucleoplasm these X-shaped structure or four-armed structures are present. These are known as chromosome. These structures are known as chromosome. Okay? But these chromosomes are not always visible in this form. In a mature cell, in an interphase cell, these chromosomes remain as thread-like structures. These thread-like structures are called, coil structures are called chromatin fiber. These are chromatin fiber. Whenever this chromatin fiber condensed, then they form this X-shaped structure chromosome. Is this clear? This chromatin fiber and its condensed form is chromosome. So, chromatin fiber is the decondensed form of chromosome. Now, I am going to show you the process of condensation of chromatin fiber into chromosome. Here, I have drawn two animal cells. In this cell, the inside nucleus there is chromatin fiber, decondensed fine thread-like coiled chromatin fiber, and here the intact centrosome. Here, the centrosome, as this cell is going to divide, so this centrosome going to divide and the centriole become separated. The asters have arranged like spindles, and inside nucleus these chromatin fibers get condensed to form X-shaped chromosome. This process is called condensation of chromatin fiber. This is known as condensation of chromatin fiber. This is interphase cell or this is mature cell, and this is during cell division. This is a chromosome, and here this is a single arm of chromosome. This is another arm of chromosome. Now we can see it is a structure. Again, this is another structure. This single structure is called the chromatid. Similarly, this is also known as chromatid, and these two chromatids are sister chromatid because they are of similar shape, similar size. That is why these two chromatids are called sister chromatid, and these two sister chromatids can join to form chromosome. Okay? Whenever chromosome separates, then it forms two sister chromatids, and whenever these sister chromatids join, they form chromosome. And this structure, this point of attachment is called centromere. Centromere. This is known as centromere. So, we can say along the centromere, two sister chromatids join to form a complete chromosome. Next question comes, what does the chromosome made up of? The chromosome is made up of DNA and histone protein. Let us see. If I drag it, then a thread-like structure, I can see here a thread-like structure. This is the chromatin fiber. These several chromatin fibers condensed to form this chromosome. So, this chromatin fiber is made up of a long thread and several bits inside it. Several bits inside it. This long thread is known as DNA. These threads are known as DNA, and these bits are known as histone. So, DNA and histone together form the chromatin or chromosome. Now, this DNA itself is negatively charged and the histone protein is positively charged. That is why they wrap one another and they form this structure chromosome. These histone proteins have 8 subunits. This histone protein is made up of 8 subunits. Now, this small unit histone with a small piece of DNA, it is known as nucleosome. Nucleosome. Nucleosome is the smallest or structural and functional unit of chromosome, because chromosome is made up of several nucleosome. Nucleosome is equivalent to a small stretch of DNA along with histone protein. As we know that DNA is the genetic material of all organisms, so we have to know about DNA in detail. We have to draw the structure of DNA and have to know what is there inside DNA, why it is considered as genetic material. Now, I am going to draw here the structure of a DNA. The DNA is made up of 2 strands which are running anti-parallelly. These 2 strands are running anti-parallelly. That means if one strand is 3' to 5', then another strand is 5' to 3'. They wound one another like a double helix. Okay. So, DNA structure is a double helical structure given by Watson and Crick. Now, this DNA is the term DNA. If we split the term, the full form is deoxyribonucleotide. So, DNA itself is a nucleotide. Now, we have to know what is nucleotide actually. These nucleotides are made up of 3 substances. One is nitrogen base, another is sugar, pentose sugar, and next one is phosphate. So, I am writing here nitrogen base, okay. Next one is sugar, it is pentose sugar, and third one is phosphate. Let us draw. Nitrogen base, there are 4 types of nitrogen base, namely adenine, guanine, cytosine and thymine. I am writing here adenine, A stands for adenine, G stands for guanine, C stands for cytosine and T for thymine. Adenine is a nitrogen base which always pairs with thymine. So, it is nitrogen base pair. Similarly, guanine is the nitrogen base which always pairs with cytosine. So, it is another nitrogen base pair. Okay. So, I have already drawn here the nitrogen bases. Next thing is sugar. In the 2 hands of this nitrogen base pair, there is a pentose sugar. And the third thing is phosphate. With the pentose sugar, there is a phosphate group. Similarly, with this nitrogen base pair, here is a pentose sugar and here is a phosphate group. So, these are 2 nucleotide sequences. Now, in between this adenine and thymine, there are 2 hydrogen bonds, and between guanine and cytosine, there are 3 hydrogen bonds. So, these nitrogen base pairs along with sugar and phosphate, they are making the steps of this ladder. Ladder means this DNA double helical structure is like a ladder, and the steps of ladders are formed by these nucleotide sequences. These all are nucleotide sequences. I can write here A, T and 2 hydrogen bonds. Similarly, G, C, 3 hydrogen bonds, like this. So, the entire DNA is made up of several nucleotide sequences. That is why DNA is called a deoxyribonucleotide. As we have already learned the term nucleotide, in contrast of this term, we have to learn the nucleosides also. Nucleosides are made up of nitrogen base pairs like this. Okay, these are nitrogen base pairs with pentose sugar only. There is no phosphate group. So, the difference between nucleotide and nucleoside is, this is nucleoside and this one is nucleotide. Difference is that nucleotide is having phosphate group and nucleoside is devoid of phosphate group. Phosphate group means energy. So, the nucleotides are storing more energy. The next term we are going to learn is gene. What is gene? Gene is, you can see here the structure of DNA and these are the nucleotide sequences. Okay. Now, gene is this specific nucleotide sequence in a DNA which can express a protein, which can code a protein, that is known as gene. So, what is the relationship between gene and protein? Actually, protein is the substance in our body that can determine any character, and gene determine our character. Gene pass on our characters. So, gene is the particular sequence of DNA. Suppose there are several nucleotide sequences are shown here, but all these sequences are not gene. Those sequences which can express itself, suppose this much sequences, here are 1, 2, 3 nucleotides. Suppose these 3 nucleotide sequences are able to express itself by forming a particular protein. So, this much portion of DNA is considered as gene. Now, this gene can carry characters from parent to offspring. So, what we have learned from today's video? What is there inside a cell? Inside cell there is a nucleus. Inside nucleus there are several thread-like structures. These are called chromatin. In a mature cell the chromatins are present. These are coil structure, and whenever cell going to divide itself, then these chromatins condense to form chromosome, and this chromosome is made up of DNA and histone. DNA is negatively charged, histone is positively charged, that's why DNA wraps histone. DNA and histone is the smallest unit, they together form the nucleosome. So, nucleosome is the structural and functional unit of chromosome. The chromosome is carrying, it is bearing genes, our hereditary unit. So, where the genes are located? The genes are located on the string-like structure, that is DNA. In DNA, several nucleotide sequence which can express itself by forming a particular protein, that much portion is known as gene. Okay. So, this is a small overview of the entire topics. Please like and share my channel, and you people stay with me.